Ladies and gentlemen, you are all welcome. So in today's class, we are going to look at the concept of peptide biochemistry. My name is Mokhtar Ali from ABU Zaria. Um, what is peptides? A peptide are short chains of amino acids that are linked together by peptide bonds. So generally, ladies and gentlemen, when we said peptides, it is actually a short chain of an amino acids. It is a short chain of amino acid, and these amino acids are linked together by a peptide bond. So we generate peptides from amino acids. So look at it here. These are individual amino acids that actually joined themselves. Look at how they joined themselves through through a bond, and the name of the bond that joins this amino acid is a peptide bond. So, and of course, when you have this amino acid, they started generating a peptide, and then of course the peptide will now further join themselves with more amino acid to generate proteins. So it means that the protein started from amino acid followed by peptides and then protein. So protein is actually a polymer of amino acids that form together by several peptides. So that is why it is important this peptide that you should understand that the peptides, they are actually a biomolecules that are involved in various biological functions. So as I said, actually for today's class, we are going to focus on the concept of peptides. What is peptide? So peptides are produced from amino acids, and then of course it is this peptide that also further join themselves to produce protein. So, and of course the peptide have a diverse biological functions, and as we are continuing the lecture, we are going to see how these peptides are important. So this peptide, understanding peptide is crucial for advancement in medicine and biotechnology. So as we are actually going through the lecture, we are going to see how these peptides are used in medicine and biotechnology. In medicine, you can use peptide in treatment of disease. In biotechnology, you can use the peptide to generate a product that is beneficial to humans, like insulin. So, what is the definition of peptide? So, peptides are typically defined as a molecule that consists amino acid of 2 to 50. So, when you have a sequence that contain between 2 to 50 amino acids, so that is what described as a peptide. So when a chain exceeds 50 amino acids, so it is generally classified as protein. So when you have a chain of amino acid that is greater than 50, so that described as a protein, not a peptide. So let's actually look at the structure of the amino acid briefly. So first, you see this is a carbon, and in this carbon there is a side chain. So the side chain is called R side chain, and there is an amino group which is NH2, and there is a carboxylic group which is COOH with also free hydrogen. So what make one amino acid differ from one another is this R side chain. Like for example, in the case of glycine is an amino acid that have an R side chain of hydrogen. So hydrogen is what reflects the R side chain in the case of glycine. And of course, look at this. This is a serine. So what is here that we have in serine? The R side chain. Look at it. This is the R side chain, and that what make it to appear as serine. So the R side chain is what make one amino acid differ from one another. So amino acid purpose. So amino acid build proteins, and the proteins are life sustaining macromolecules. So the amino acid, they are the building blocks of proteins. And the proteins that we are talking about is actually the life sustaining macromolecules. We are alive, we are active, we are healthy, we function, or we do all life processes, activities because of the proteins. So once you have, because proteins is what determine the function of each and every cell in our body. Like for example, the red blood cell is responsible for the transport of oxygen because of the hemoglobin, which is also a protein. The brain that we have, actually, it is responsible for cognitive function, memorization, calculation, and so many other things is because of the neurotransmitters that are there. So and those neurotransmitters, some of them are proteins while some are peptides. Okay. So when the chain, as we said, exceeds 50, so it's classified as a protein. So peptide vary its length in structure and function depending on the amino acid composition. Because in a peptide, we have different classes of amino acids. We have 20 different amino acids, and these 20 amino acids are used in generating different peptides and different proteins. So the length, as well as the structure and function of each and every peptide, depend on the amino acids that are used in building such peptide or protein. Because there is a different permutation and combination of these amino acids that make hundreds, thousands, and millions of proteins and peptides. In terms of their structure and functions, it also depends on the composition of the amino acids. So, structure of peptides. The peptides have a backbone composed of repeating amino acid units. So it is a repeating amino acid that composed to produce a peptide, and each amino acid has a distinct side chain that influence peptide function. So each of the amino acids has its own side chain. Like for example, look at this. In the case of arginine, the R side chain, this is the R side chain of arginine. In the case of histidine, this is the R side chain of histidine. All this complex structure here is the R side chain of histidine. And of course, in the case of arginine, this is the R side chain. In the case of lysine, this is the R side chain, all from here. And then in the case of aspartic acids, this is the R side chain from here. So you see each and every amino acid have its own R side chain, and that is what make them differ from one another. But they have serine, threonine, asparagine, glutamine, isoleucine, leucine, methionine, phenylalanine. All these are different amino acids and they have their different R side chain. So the sequence and the arrangement of these amino acids is what actually determines the peptide properties. So you should understand the arrangement and the sequence of the amino acid is what determines the peptide properties. So what are the types of peptides? So peptides can be classified into various categories. We have hormones. We have peptides that are hormones. We have peptides that are neurotransmitters. And we have peptides that are antimicrobial in nature. So each of peptide has a unique roles and mechanisms of action in the body. So understanding these categories help in studying their functions and application. So let's actually look at the examples of the peptides that are neurotransmitters. You see adrenaline, noradrenaline, dopamine, serotonin, glutamates, endorphins, GABA, and acetylcholine are examples of neurotransmitters that are peptides. So let's start with adrenaline. So adrenaline is a fight or flight hormones. So this adrenaline is usually produced in stressful situations, which increases heart rates and blood flow, leading to physical boosts and heightened awareness. So you see, adrenaline is a peptide and also at the same time a neurotransmitter, and it has functions of increasing heart rates and blood pressure, which actually change someone's mood for fights or flights. And we have no adrenaline, so it affects attention and responding actions in the brain. It also contracts blood vessels, increasing blood flow. So this is actually some, and of course we have dopamine, which is a pleasure neurotransmitter. So feelings of pleasure, also addiction, movement, and motivation. People repeat behaviors that lead to dopamine release. So dopamine is actually a pleasure hormone, and it's also an addiction hormone. It's actually help in movement as well as motivation. We have serotonin, which is a neurotransmitter that actually bring a very good mood. So it contributes to well-being and happiness, and it also helps sleep cycle and digestive system regulation. So it affected by exercise and light exposure. So that's why in many cases it is always better when you are going to sleep off your light. The reason is if you don't off your light, then your serotonin levels will be affected. And once your serotonin level is affected, it means that you are not going to have good sleep. Because if you sleep, you need a good mood. You need a well-being and happiness while sleeping. So, but if you didn't off your lights, the serotonin level will be decreased. So therefore, you will not enjoy your sleeping. And we have GABA, which is a calming neurotransmitter. We have acetylcholine, which actually helps in learning. It's involved in thoughts, learning, and memory. It activates muscle action in the body. It's also associated with attention and awakening. We have also glutamate, which is also a memory. That is the most common neurotransmitters involved in learning and memory. So some of us, we are not actually intelligent because of the low secretion of these neurotransmitters like acetylcholine, glutamate. So if we have decrease in the concentration of these neurotransmitters, it will definitely affect our learning process. It will definitely affect our learning process. So that is why we always encourage people that have that doesn't have a good learning memory to always engage in taking foods that are rich in these molecules like the acetylcholine and glutamate. So if you can be able to identify food like, for example, banana, it is rich in glutamate. In fact, even the Maggi that we are taking, we have sodium glutamate. So the glutamate there for taking proteins well, of course, it can lead to producing glutamate, which can help in learning and memory. It also regulates development and creation of nerve contacts. Then the next one is endorphin, which is euphoria. It released during exercise and excitement and sex, producing well-being and euphoria, reducing pain. So actually, the pleasure hormones or the neurotransmitters is what actually bring excitement as well as well-being during sex. So it's what make us to enjoy sex. If you enjoy sex, is because of these endorphins. So it depends on how you produce these endorphins toward your sexual partner. So that is why people have different attraction towards their partners. So understanding these categories, it can help in studying their functions as well as application. So with all these things that we are saying, you see that these are neurotransmitters and at the same time they are peptides. So they are, but in the case of glutamate is not a peptide, it's an amino acid. But endorphins, dopamine, serotonin, norepinephrine, and adrenaline are the examples of the peptides. Okay, so peptide synthesis. So peptide can be synthesized naturally by ribosomes through translation. So you see, remember from basic biology, the protein synthesis takes place in the cytoplasm of the cell. So therefore, even the peptides, because peptides are also like the starting raw material for protein synthesis, so it also synthesized in the ribosomes. It can also be artificially created. So you can create peptide artificially using a chemical synthesis techniques. An example of the chemical synthesis techniques that you can use to artificially synthesize this peptide, we have solid phase peptide synthesis, SPPS, is a common method used for laboratory synthesis of peptide, and this is how it is. Peptide synthesis resin bit. So this is how it is. Then you have a peptide linker, you have a growing chain amino acid. So this is actually a brief description of how the peptide are synthesized, and the method is solid phase peptide synthesis, which is a laboratory technique that is used in the synthesis of peptides. Now, what are the biological functions of this peptide? So peptides play important roles in signaling. When we said signaling, it means that they are very important in sending message across the cells. It also involves in regulation, that is the control particular biological processes on or off a particular biological process that is taking place in the body. So you can use peptides. Peptides are used in either off or down regulation, that is to increase or decrease the biological process in the body. And some of them, they are catalysts, that is, they can increase the rate of the reaction. It means that some of them are enzymes, so they can act as hormones. So some of the peptide can act as hormones, influencing physiological processes such as metabolism. Like for example, there are millions or there are thousands of metabolic reactions. When we say metabolism, it means the totals of biochemical reactions or biological processes that are taking place in the body. And of course, some of these metabolic pathways, they are activated by hormones like glycolysis, lipogenesis, glycolysis, glycogenolysis. All these pathways or all these metabolic pathways are controlled by hormones, and the hormones are peptides. So that is why, ladies and gentlemen, understanding the concept of peptide is very, very important, because if you are able to understand the nature of how the peptide looks like, it will definitely help you to understand that the peptide as well as the proteins, they are not the same, and what makes them different is because of their different amino acid composition as well as their arrangement. So neurotransmitter peptides, they transmit signals in the nervous system affecting mood and behavior, as we described previously in the previous slides. So peptide in medicine. What are the applications of peptide in medicine? Peptides are increasingly being used in drug development. And of course, I know that there are so many scientists that they can use peptides to actually inhibit a particular enzymes in order to develop a drugs due to their specificity and effectiveness. So you can use peptide as a drugs. Yeah, you can use peptides as a drugs. So that is why, because of their effectiveness and specificity. So some scientists now, they use artificial intelligence to generate peptides and target a particular protein that is essential for either a disease pathogenesis or for the development or survival of a particular parasite. So if you can design or identify peptide that can inhibit such proteins, then it can serve as a drugs. They can be designed to target a specific receptors, minimizing side effects. So you can actually design a peptide that can inhibit a particular receptors. So examples of this peptide include insulin peptide for the diabetes management. So the peptide insulin is also an example of peptide. We also have peptide vaccines for immunotherapy. So can use peptide as a vaccine in order to control diseases or in order to manage diseases that are associated with immune system, like cancer. Cancer is also a disease that are associated with immunity. So you can design a peptide that can actually control the immunity that is associated with cancer. Of course, you can cure cancer. So peptide hormones. So you see here we talk about peptide in medicine. So now peptide hormones. So peptide hormones such as insulin and glucagon regulate metabolic processes. So you can use these hormones to control or regulate biological processes that are taking place in the body. You see, these are water-soluble hormones. So water-soluble hormones, once they actually get into the cell, then they will bind to the receptor. So once they bind to the receptor, it will now activate a receptor. It will activate the receptor called G protein coupled receptor. It will be activated, which in turn activate adenylyl cyclase, and then the adenylyl cyclase will now convert ATP to cyclic adenosine monophosphate. And the cyclic adenosine monophosphate will now activate protein kinase. And once activated, then the activated protein kinase will now activate protein kinases. So, the protein kinase will now phosphorylate the protein, and the phosphorylated protein, depend on the types of the protein, it can be active or inactive. So this is just a signaling pathway, and it is just to show you that using a peptide hormones, we can actually control or regulate metabolic processes like glycolysis. As we said, this is how it starts. The water-soluble hormones first will now bind to a G protein, which actually leads it to its activation, and then the activated G protein will now activate adenylyl cyclase, and then the adenylyl cyclase will now catalyze the conversion of ATP to CAMP, which is a secondary messenger. And then the secondary messenger, which is CAMP, will now activate protein kinases, and then the protein kinases will now phosphorylate proteins in the cytoplasm, and this activates proteins, allowing them to alter cellular activities. So this is actually the work of peptide hormones. So from this pathway, this is how the signaling pathway is described. So you can use a peptide in signaling pathway. So they are produced in specific glands. The peptide are specific, produced in a specific gland, and then now released into the bloodstream, where they are going to be transported to their target organs. So these hormones also bind to receptors on the target cell, triggering physiological responses as we explain here. So it triggers the physiological responses. So neuropeptides like endorphins, they modulate neural activity and pain perception. So they are involved in the variety of functions including mood regulation and stress response. So we have neurotransmitters. An example of these neurotransmitters, we have endorphins, which are involved in modulating neural activity as well as the pain perception. And in addition, like endorphins, it is involved in mood regulation as well as the stress response. So if you are stressed, and also there are some hormones that need to be released when you are stressed, like adrenaline. But in this case, endorphins is an example of neuropeptides that involve in mood regulation and pleasure. So understanding this peptide, it will open avenues for treating neurological disorder. So some people, they are stressed and they are in bad mood because of the lack of these neuropeptides, neurotransmitters. So if you can actually produce artificial neuropeptides, you can actually administer it into the patient and change the perception as well as reduce the stress and improve moods condition. So this is actually the pathway of pain perception. It started here. Like for example, if you are now maybe the pain perception may be something like null. So the first thing, there will be nauseous stimulus. So the nauseous stimulus will now actually transduce at actually peripheral nociceptor. And then of course the transactions, of course, so the message will now transmit to the central nervous system. So you now have pain. So as a result of this neuropeptide, it can modulate and control the pain perception. So this is actually what we want us to, and what we wanted to understand. If there is a pain, there is always a communication between the brain. So this pain perception is actually modulated and controlled by the neuropeptides. So then antimicrobial peptides. So there are a series of peptides that can be used or that are part of innate immune response against pathogens. So they usually actually improve or boost innate immune response, which actually clear pathogens in the body. They have broad spectrum activity against bacteria, viruses, and fungi. So you can use them in the control of the microorganisms like bacteria, viruses, and fungi. So that's why a lot of scientists now and the researchers are working towards to develop antimicrobial agent or antimicrobial compounds from peptides in order to treat in order to treat pathogens. So research in antimicrobial peptide is promising for developing new antibiotics and amid raising resistance. So currently we know that there are a lot of resistance of there is a lot of resistance of antibiotics by microorganisms. So now researchers are working towards using these antimicrobial peptides to reduce the challenges of drug resistance. So this is actually one of the work conducted by some scientists by improving antimicrobial peptide classification with deep learning-based prediction. So using an artificial intelligence, you can actually improve antimicrobial peptide class. In fact, even in terms of their activity. So you can start from in silico using a deep neural network as well as the fine-tuned natural language processing to actually translate it or train. So this is actually because at your own level, we don't need to go deep into this. You can use deep learning as well as the artificial intelligence in identifying as well as developing new microbial or antimicrobial peptide for the treatment of disease, especially microorganisms. So peptide therapeutics. So peptide therapeutics are drugs derived from peptide and are designed to treat disease. They can be engineered to improve stability, absorption, and availability. So you can use these peptides to treat diseases. So that is the peptide therapeutics. We have some peptides that are used in the treatment of diabetes, treatment of some microorganisms. So we have so many of them. So you can engineer some of these peptides to improve their stability, absorption, bioavailability, and so many other things. So the peptide-based drug market is rapidly expanding due to their clinical success. So peptide therapeutic market, the global peptide therapeutic market is led by a key players like Teva Pharmaceutical Industry, AstraZeneca. So these are all companies that are actually leading the research in drug development in the area of peptide chemistry. So peptide vaccine. Peptide vaccine also utilizes a specific peptide sequence to elicit immune response. So there are peptides that can actually inject into humans, and then of course it will elicit immune response in order to actually cure or prevent, in order to cure or prevent a particular disease. So the aim of this peptide vaccine is to stimulate the body's immune response to recognize and attack cancer cells. So you can use peptide bonding immunotherapy to treat cancer. So this approach is being explored for various diseases, including infectious diseases and cancer. So you see, you can use a peptide vaccine in the management of infectious disease and cancer. Then peptide in biotechnology. So in biotechnology, peptides are used for various applications, including diagnostics and therapeutics. So they are serve as a biomarkers for disease detection and progression also. So peptide-based assays and therapies are evolving fields with significant potential. So these are the applications. So you can use this peptide in diagnosis of the disease as a biomarker. You can also use this peptide in animal breeding agriculture. You can use them to improve food technology. Like for example, you can develop insecticides from peptides that can actually kill, as well as diagonal, as well as preventing insecticide from causing any harm on our plants. So you can use it in improved food technology, fermentation, and so many like recombinant DNA technology. All these are the field where you can use these peptides in biotechnology. So what are the challenges in peptide research? So peptides often face stability issues in biological environment, limiting their use. So delivery method for peptide can be challenging due to rapid metabolism and clearance. So ongoing research aims to improve these obstacles and enhance peptide utility. So ladies and gentlemen, the aims of this lecture is to give you an overview of the peptides as well as their application. So thank you very much for listening to this lecture. And if you have any questions, you can ask your question on the comment section. And I hope the video will be of help. And if this is the first time, please try to join the YouTube as well as subscribing the YouTube channel. Thank you very much.